All right, good. Let's start. Continue recording. Hello, everybody, and welcome to our lecture. So as we had it before, our two TAs, Vanessa and Matteo, will be online and visible, and then they'll take questions from the chat, and then you can interrupt me if you want, Matteo and Vanessa, and the lecture is being recorded, so you can also watch the recording later again. Okay, good. So today we talk about the neural code, from temporal to rate coding, from single neurons to networks. And especially this topic, temporal to rate coding, has been discussed very intensively in the last, I would say, 10 or 15 years, right? What is it that really neurons use to encode information? Is this more a temporal code where the specific time of a spike is important? Or, for example, this is a rate code, right, where the stimulus is only encoded in the frequency, in the firing frequency of the neurons. And then the second big question that we'll look into today is basically, how do single neurons encode information? But it's clear that, if we would rely everything that we do, all our information processing, just on a single neuron, right, single neurons fail, they're variable, then we wouldn't get far. So we need to think about how populations of neurons encode information. Okay, these are my favorite books. If you want to take a look, a couple of them you can also find as PDF online, I think almost for free. There's the spike books, which is really about spikes and single cells and how stimuli are encoded and how you can determine the probability of the spike given a certain stimulus. Then in the middle we have the classic, the Diane and Abbott Theoretical Neuroscience. Also a lot on the single neuron level, talks about basically how neurons change their firing frequency, what input-output function they have, and so on and so on. Also a very cool book. I highly encourage you to take a look into this book. And then if you're more into pattern recognition, machine learning, with a little bit ideas from neuroscience, and this Bishop book I think is really cool, especially if you're like in neuroscience, and if you look for methods to classify, to find your input-output function of your neuron, to find a decoder or classifier. Okay, good. These are the papers and the reviews that I included in the lecture today. One very inspiring one has been from Romain Brett. We'll talk about this in a couple of seconds. Shams and Zader wrote a nice review about temporal versus rate coding. Perkel and Bullock is the classic paper, basically first time suggested the idea of coding as neural coding and the causal relationship. Then we have the Quiroga paper, where very famous paper where people have looked at monkey high visual cortical processing and found cells that respond to faces. The classic paper on neural code is Georgopoulos, where they look at the monkey reaching task. Then, for example, my paper where I look more in towards population coding. Good. Okay, so let's dive right in. How can we think about coding, right? And in general, coding, think about you have an emitter, right? That's, for example, you have a message, and then you encode this in some type of form, right? You transform this information, for example, into a Morse code. And then you send it out, and then there's a receiver, right? And he or she would need to know how the message is encoded, right? To actually make sense of it, right? To reconstruct the message or the sentence or whatever that you encoded. And kind of we can think about this in a similar manner also what happens in the brain. So you have a sensory signal, and then when the sensory signal enters your retina, right, you have rods and cones, and then you have ganglion cells, and at the level of ganglion cells, we're only at spikes, right? So already in the retina, your signal is encoded in spikes, and then when you measure the representation, the response to a visual stimulus, for example, in the primary visual cortex, right, you have more complex spike patterns, right, that you might or might not understand. And then, of course, if another higher level brain area wants to make sense of the sensory stimulus, right, it needs to encode somehow, right, what information, what stimulus signal is represented by the activity signals in the primary visual cortex, right? And us, if we measure in the primary visual cortex, we need to think about, okay, how can we get, right? How can we basically decode, right, from the spiking signal, the sensory signal again? Now, there is a certain, direction for encoding and decoding, specifically for the brain. The idea is somehow that the stimuli, right, or that a visual stimulus has a causal relationship. So the visual stimulus, right, elicits somehow a code in our primary visual cortex, right? And this code we can, for example, decode, right? And then we reconstruct, for example, our input, right? So it goes into one direction, it's causal, it doesn't go into the other direction. Good. And this is also kind of, discussed long in the Perkel and Bullock paper. It's called the problem of neural coding, is to elucidate the representation and transformation of information in the nervous system. Good. Okay, so it's about representation and information. So I wanted to show you this. This is also from Romain's paper. I thought it's a pretty cool example of how one could think about neurons and how they actually encode things. So this is a single cell organism. It's called the paramecium. And this is, if you so will, it can actually spike. So, Romain Brett calls it a spiking, a swimming neuron. And if you look at B in the right plots, it has these little flagella around it, right? And these little flagella are somehow, they're moving all the time, right? But they're all chaotic, right? So in the normal state, this paramecium wouldn't go anywhere. No, what's its goal? Its goal is what you see in A, to find some position, some location where you have a high concentration of a certain chemical or nutrients and so on, right? Now what it does, and this is pretty cool, it takes somehow this gradient, this information of, let's say, a chemical, and then somehow translates this, right, into an intracellular spike. So where the concentration gradient is the highest, right, and this inside the membrane of this paramecium, a spike is triggered, and this spike then travels around this membrane, right, along both directions, and then synchronizes these flagella in a way that this paramecium swims towards this higher concentration gradient where it's happy and where it gets all its nutrients and chemicals. So you can think about now, how does this organism transform information, right? It transforms outside information. This is our concentration gradient into an internal signal. This is an action potential that has a certain effect. And this effect is that it swims towards this higher concentration gradient, right? So think about that neurons, right? And I see neurons also as, in a similar position. Single cells and possibly also neurons encode information so that they reach some other state, so that they can really do something useful either for themselves or for the organism. That's how I think we should think about coding. Good. Okay. Romain Brett talks about three elements of coding: correspondence, representation, and causality. And thereby correspondence, you can think about it as correlation between two domains, right? If I have visual signals and they elicit spike trains, right? We call this a relation code. And one could, in principle, right, reconstruct, that's the idea, right? The original message from the encoded methods. That's the correlation. Now, we have to be careful here, right? Because correlations doesn't mean that it's causal, right? So not all cases of correlations are causal. For example, you would never say that the spike train elicited a visual signal, right? Or something here. Climate scientists, for example, really ask how rain encodes atmospheric pressure. Good. Okay. Yes, and this is already kind of yeah. So we would not say that visual signals encode retinal spike signals. So this causal relationship, this unidirectional property, is really important for, at least for the neural coding. Okay, so just to define what I mean when I say encoding or decoding, right? So encoding defines the process of how a stimulus causes a pattern of response, right? And we can think about it, the brain we're building, right, or we are probing a proximate mechanistic model of the world, right? So the idea is somehow that our brain encodes or has internalized all these stimulus in its own code, right? And the form of encoding, when I learn to encode a stimulus, I build an internal model of the representation of the other world, of the information of the other world. Now, decoding, right, is the idea of when I want to decode either, right, we have many brain areas in the brain, right? So for example, the hippocampus needs to decode the information that is currently present in the primary visual cortex, right? Or if we are neuroscientists, if we stick an electrode in the brain and we measure activity in the primary visual cortex, then the question of decoding would be, how can we reconstruct the stimulus, right? The outside information from the activities that we measure. Okay, good. So the task about finding or learning an encoder and decoder is basically about finding the probability here of a response given a certain stimulus, that's our encoding, or finding the probability distribution of a stimulus given a certain response, right? Good. Now, what is and this is the magic question in neuroscience, what's the relation between a stimulus and a response, right, in the brain? And if you probe, for example, a neural response, and you could just measure from a single neuron, then what you often see is something like this, right? So you have the neural response on the y-axis, and this is usually firing frequency, right? Or if you look at calcium imaging, it's fluorescence trace, peak amplitude, or something else. And then the y-axis, you have stimulus parameters. Now, what you see is that with varying stimulus parameter, right, you get some type of variations in the neural response, right? And here, obviously, we have no, clear preference for this neuron to the stimulus parameter. Good. So the first experiment that explored this, I think, and brought up this idea of tuning was Georgopoulos in 1982, at least for the motor system. And in this experiment, what basically they had a primate, a monkey, sitting on this desk that you see in A. There was this robotic arm that the monkey could move, right, in 8 different directions. And then he had a task to do, right? So he did these movements, he got a little reward. And pretty much after a while, he learned very stereotypic movements to all these positions, right? From 90 to 0 to 170 degrees and so on and so on. Now, what the group around Georgopoulos had done at the same time is they have recorded activity from the monkey motor cortex. And what they found is something like this. And this is the plot from the original paper. This is a motor neuron that they recorded from. And what you basically see in the top plot is the spiking, right, of this neuron, and the lines are the individual trials. And then what you see is that this neuron seems to spike when the monkey is reaching in one certain direction, right, around 140 degrees, but not into the other, right? So here we have a neuron. It's even less, right? It stops firing. So it goes like below baseline. So here we have a clear kind of tuning curve. And this tuning curve basically somehow this neuron, right, encodes basically the parameter that this neuron encodes is the reaching direction. Good. And this type of like Gaussian-like tuning curve is pretty classic, what you see in many other brain areas too. Good. Another experiment which I wanted to show you is this one here, very classic for those of you who have not seen it, is from Hubel and Wiesel, even earlier in the 1960s, and they have recorded in cats. And they basically have, found how visual stimuli, right, either natural or more complex, are encoded in the brain. I'll just start this movie, and then I hope you can hear it.
The researchers actually listened in to individual nerve cells firing in the anesthetized cat as they presented it with different visual images.
When we started working, Torsten and I, in the late 50s, we set up our first experiments, and they didn't go well because at the beginning we couldn't make the cells fire at all. We'd shine lights all over the screen, and nothing seemed to work. And rather by accident, one day, we were shining small spots, either white spots or black spots, onto the screen, and we found that the black dot seemed to be working in a way that at first we couldn't understand until we found that it was the process of slipping the piece of glass into the projector, which swept a line, a very faint, precise, narrow line across the retina. And every time we did that, we'd get a response.
All right, good. Very short movie is one of my favorite ones because it really shows how neuroscience research has been done in the 1960s. And a very famous work came out of this. So question for you, people who haven't seen this video, what is the cell? You hit the cell, right? The clicking cell. What's the cell in primary visual cortex that Hubel and Wiesel measure in this cat responding to? And I just throw this question out in the Zoom cloud and then I'll resolve it in like 10 seconds or something. Motion. Yeah, that's, yeah, it goes to the right direction, yeah. Motion of this dot or what type of motion? Of the dot, yes. Okay, yeah, good. Edge filter plus motion filter. Yeah, that's good, yeah. All right, yeah, so I can resolve it. So what this neuron, and this was almost, you could call it, half an accident. What they found is that they had this slide, Hubel and Wiesel, right? This transparent sheet, right? And on this transparent sheet was this dot. But when they slided the dot through the picture, there was a slight edge from the sheet, right? That also moved through the picture. And what they found is what the neuron responded to was not the movement of the dot. Namely, it was the fine edge, right, that swept through the field of view, which seemed to have excited the neuron. Right? So here, let's go to the next. Okay. So what they found is basically orientation and direction selective neurons, right? So direction would be somehow with motion, right? Orientation would be without motion. And this is this classical experiment. So they had the cat that recorded using an electrode. The cat was looking at the screen, right? And then they kind of changed this experiment. They basically just had these bars, right? Which is a certain, call it an edge or something, that they moved through the visual field of view from the cat. And then they measured the neuron's response. And here is an example of a neuron, right? On the right, you see, examples of the neural response given certain sweeps in certain directions, right? And in B, you see the tuning curve of this neuron, right? And here it's basically 0 is the preferred angle of this neuron, is somewhere between, 45 degrees or something, right? Yeah, and then the firing rate goes up from 0 hertz to about 50 or 60. That's quite classical. And again, we have a neuron that encodes here not the reaching behavior, but it encodes, right, with a spike rate. It encodes the direction of this little edge filter moving across the field of view. Good. Okay, and this is it again. Here, kind of, this is another study. It's actually from a textbook where you see a neuron that's highly tuned to the vertical direction, and kind of course, when you look around the primary visual cortex, you find neurons that are differently tuned to all types of these directions here. Right? Good. And this is very consistent with more recent work, also in primary visual cortex. What people did here is a little different approach. They didn't sweep things to the image. They basically showed the primate natural images as what you see in A, right? And then what you can do is you can measure the neuron response to every of these images, right? And then make some type of template or average, right? And figure out, okay, what's the neuron maximally responding to, right? What feature in all of these images? And what you get then is something, as you see in C and D, if you look at these two gray images, right, is something kind of a filter. And we talk later about, how we can think of neurons as spatial and temporal filters. And what you see is that, for example, in V1 and also in V2, you seem to have these edge filters, I call them, right, with somehow, a white, this means a high intensity and then a suppression region around it that have a certain orientation or a certain direction, right? So this is kind of basically how visual information is encoded in the brain on the lower levels of cortical processing, right? What we know, of course, that there's a hierarchy, and we know that there's not only primary and secondary visual cortex, there's, up to the fourth, V4 visual cortex, there's kind of IT cortex, there's AI cortex, so there's much more. Can't see your cursor. Okay, I don't have a cursor. Good. So this study recently fascinated me, where people then, went into higher order cortical hierarchies. And this is a study from James DiCarlo from MIT. and basically figured out, okay, what type of features, right, do these higher order cortical cells in primate visual cortex encode? What do they maximally respond to? And to figure that out, they did a super cool experiment, which is basically, first they trained a deep neural network, what you see here on the top, to recognize images, right? In this case, different images. I think it actually was it ImageNet? I need to look it up again, which set it was. It doesn't matter so much. So this network learned to distinguish, for example, dogs and cats, right? And then it had several layers. And then what you could do is, right, you could go to layer 4 of your visual network, and you could design easily a stimulus, right, that triggers individual neurons, or maximally triggers individual neurons in layer 4. So then what they did is they took these specially designed stimuli. That's what you see on this kind of artistic image on the lower right. And they showed them to the monkey. And what they found is that when they measure in higher order cortical areas, they could maximally excite neurons in the monkey IT cortex, right? So they had basically found the tuning, right? The feature that these neurons somehow encode from visual data, which I thought was pretty cool. And obviously, this is only shown for the visual pathway, but you could extract and do probably similar things with the auditory pathway, right, with the motor pathway, with somatosensory pathways, if you go along the hierarchy from low-level information processing to high-level information processing or encoding. Good. Okay, good. So now I told you what happens here, right, in V4. And then you see these other areas, AIT cortex, for example, right? So there's a couple of even higher order areas in the primate, in the monkey. And this is what the study of Quiroga et al. Have looked at. How do these areas encode visual information? And what they did is they showed the monkey images of people, and then basically measured the response of these of neurons in the MTL cortex here. Good. And what turned out, right, very simple, if you just, then look at which pictures are those, right? And this is kind of also a reason why the study is very famous, like it or not. The monkey doesn't know any of these persons, right? So here we had a neuron that responded to all the pictures from Jennifer Aniston. And when they then looked, for cells that were also, highly excited, but not super excited, these cells, for example, right? This is another cell. Then they found this cell, and then again you can look at which pictures are eliciting this response, this high number of spikes, and then you find a cell that kind of has Jennifer Aniston and Brad Pitt in there, right? So yes, was also in the news, this paper, because of actually this finding. Good. But this shows you somehow that a neuron, then if you go even higher level, neurons are maximally activated, right, by in principle high level concepts, objects, people, cars, buses, whatever you like, right? So that's what activates them, and that is what they directly encode, right? Versus on the lower level, if you go very back to primary visual cortex, we have a neuron really responds to these edges or directions or orientations in the field of view. Okay, good. So yeah, and as I said, this is for visual cortex, right? But you could do the same hierarchical idea and encoding for auditory cortex for other modalities, of course. Good. So I would like to, from here, transit a little bit to another type of encoding, as it is has been observed in rat hippocampus, where neurons somehow observe or encode a place, right? That's why these cells are called place cells. Then there's also grid cells, which we talk about later. And for the discovery of these types of cells and for this types of encoding, these three people here have gotten the Nobel Prize in 2014, John O'Keefe, a good friend of mine, super cool guy, and then the Moses, who have mostly, more investigated place cells, but also investigated more grid cells. Good. Okay. So how is information encoded? And here we are not in the cortex anymore, we're in the hippocampus. And I actually jumped, so this one first. Good. So here we are in the cortex, and on the left you see a rat brain, right? Much smaller than the monkey or the human brain. And this orange part should basically kind of annotate where the hippocampus is, but keep in mind, it's not on the cortical surface. It's really inside the brain. It's subcortical. It's below the surface. Right? Good. And obviously also we as humans have a hippocampus, and we also have a medial entorhinal cortex. Good. So this picture on the right is a figure I copied from the original paper from John O'Keefe, where basically the first time he reported these place cells. And so he recorded in this rat the activity of this neuron. And what you see is if the rat is at place A or B-ish, right, then the cells give a high firing, right? Whereas if the cell is in place E or some other places, the cell does not spike at all. Good. So people, of course, have, now since 20 years investigated hippocampal place coding. And this is a cool experiment that I wanted to show you, where basically there's a recording from a rat, and the rat has actually learned this maze here, and it's now going along this track. And what you'll hear is this little click, click, click. These are the neurons in hippocampus firing, right? And what you'll see is that there's certain neurons, right, that are firing at certain places in this maze, right? And because we do some spike sorting, not explain exactly what this is, we can really assign each click to different neurons, although they sound very much the same. You see there's a dark blue place field right around the first corner. What you only can guess here a little bit is that a cell can have multiple place fields, right? It's not just that one cell has one place field and then that's it, right? So you see, for example, the light blue cell active multiple times, the dark blue cell, the turquoise cell, so they all have one, sometimes two, sometimes three place fields. Okay, good. That was it. So this is kind of the summary here of this experiment. And by doing this, of course, right? So now we can ask, how is space, how is this whole maze encoded in a population of hippocampal neurons, right? All these colors here represent the spiking activity of different neurons. Then what we can do is we can basically learn, for example, a decoder, right? And that's what we did here in 2013. So this was, I just started my postdoc in this lab, and then my friends published this beautiful paper where they basically recorded from multiple neurons in the hippocampus while the mouse was exploring a track, a linear track in this case also. And then what you can do is you can train a simple classifier, right, that looks at the cells, which cell is spiking, right, and then predicts which position is the mouse in, right? And we did this and could actually, just by looking at the hippocampal neural activity, we were able to basically decode the movement or decode where exactly the mouse is at the position and where it will go. Good.
So there is a question about the, I think, slide before. So 2 slides before. Does the distributions of neurons on the left graph represent their location in the brain?
No, this is some
left graph. I think you have to go back to see the left graph.
Yeah, okay, good. I'll try. Yeah, I don't want to forward the movie.
I'm guessing it's the overall versus selectivity.
Yes, it's this here. Yeah. So no, on the left, what you'll see is basically what we record is spike waveforms, right? We record an electrical signal of this spike. This is also what you hear in the audio monitor. And these spikes all look similar, but they have some fine different nuances. So what we do is spike sorting, right? You push somehow the form of the spikes, you can do some type of dimensionality reduction, project these spikes into a space where you can separate the different neurons. Right? So here, think of it, you would do principal component analysis, you would have principal component 1 and 2 on these two different axes, right? Yeah, it's for spike sorting. It has nothing to do with the position in the brain. These cells are all in hippocampus.
And a very quick thing, maybe you can turn on your pointer. Just
yeah, I don't have a pointer.
okay. So if not, just say top, bottom, left, right. Another question: how were those neurons recorded? Does the technique record several neurons at once?
Yes, I'll have a slide on the technique. So there's two methods. The first is tetrodes. It's like a four electrodes in parallel that you implant in the brain. And then there's a new cool method called pixel probes. I have a slide on those, show you how they look, where you have up to 300 electrodes simultaneously in a shank that you can use for recording. And you can usually record a couple of hundred neurons simultaneously with that in the mouse brain. Yeah, I'll show you in a couple of slides. Good. Okay, so what we talked about so far all was kind of rate coding, right? So you've seen that somehow a stimulus, orientation, direction can be encoded in the frequency of firing, and that this usually has some Gaussian-like shape. But there's another way how you can encode information. Let me say that so far, I think most of the brain areas you find definitely rate coding, right? You find that your stimuli are encoded in the rate, but here and there are some special brain areas that encode somehow information in the timing of spikes, right? Good. Okay. And this is called a temporal code. And there's different versions of a temporal code that we'll not all discuss, but the main ones here. So the first idea, right, and I start with the very left, is time to first spike. So if you present a stimulus and then to whatever the mouse, and then you would wait which neuron, right, is responding the fastest, which neuron has the shortest delay time, then you could say, well, this neuron is the most tuned to this stimulus, right? And the neuron that responds the latest is the least tuned to the stimulus. And this would be a time to first spike encoding, right? So it's not only fires one spike, but you log how much delayed it is. So the reference signal here for our spike is the stimulus, right? What you could also do is, of course, you could look at synchrony. And for the synchrony, you need at least 2 spikes to occur, right? And either from 2 neurons or from the same neuron in time. And then you could look how synchronized, right? How close in time are these spikes? And then you could say, well, if the synchrony of all the neurons, for example, is very high, right? Then the neuron is very responsive to the stimulus. If the synchrony is very low, right, they all spike at different times, then it's very, the stimulus response is very low. The last possibility to encode via temporal modification information is phase precision or phase coding. In hippocampus, we just saw an example from hippocampus. What you see is that there's a background oscillation in neurons, right? So this is usually mediated through inhibition, where the membrane potential of hippocampal neurons continuously fluctuates, right? We'll have a couple of slides that show you with a theta frequency. And then depending on where the cell exactly in this oscillation phase spikes, right, encodes, for example, the position of the rat or the mouse. Good. Okay, so I would like now to show you a couple of examples of these temporal codes, and then we also have a whole temporal and rate code quiz. Okay, good. Okay, good. So yes, these are just the definitions. Rate coding refers to information carried by the firing rate. This is mostly, I think, what you find in the brain. And then temporal coding, right? You have these 3 different ideas, either through synchronicity, through phase coding, or through time to first spike. Good. Okay, this is just from my old work 10 years ago. I was actually into that when the discussion about, how is information encoded was really a big discussion. Is it a temporal or is it a rate code? And what I wanted to know is basically, what are the different effects intracellularly for this pyramidal neuron on calcium influx and so on, backpropagating action potentials from the soma down in dendrite? when you have a temporal or rate code, right? And this is a nice example to show you what I mean with temporal code and rate code. So a rate code would be, I have a stimulus, right? I inject a certain pattern into these cells. And if you think about the whole stimulation period, the number of APs is always the same, right? But the frequency was different. And the top row, this was actually pattern that we had recorded in vivo in these cells due to stimulus presentation. And the bottom row was just us kind of turning this temporal code into a rate code that has the same amount of action potentials. Good. And we found that these two different rate and temporal codes actually had very different effects intracellularly on the cell's calcium, on backpropagation action potentials. Good. Okay. So I would like to make a short quiz with you, and then I come back to phase precession and other temporal coding methods. So here we have recorded neural responses in the cat Algian. That's the visual thalamus of the cat. And they had seen the stimulus, right? And you could think about this as, some variable, some intensity of a pixel or something going up and down. And then they had done 2 experiments in which they have basically recorded the spiking activity of these neurons. And then on the y-axis, you see the trial, right? Good. And the question here is this a temporal or rate code? And I'll give you just a couple of seconds and then I'll resolve this. Okay, good. So this is the idea that this here is a rate code. And what are these neurons responding to? It seems they're responding to, ons and offsets, right? Strong onsets and offsets of this stimuli that you see on the very top. So if this goes down, right, very strongly, then you see something. If this goes up, then you also see something. Okay, good. Let me move on to this example here. So this is another experiment. Is this a temporal or rate code? So here, this was work in primates. And you have this, the primate is watching these moving dots, and they either have 0% coherence, meaning they all move in different directions. They're 50%, then 50% of them move into the, into one direction, the upper directions, or you have 100% coherence. And then on the bottom, you see basically during one trial, right, or doing multiple trials over 2 seconds, you see the activity over during these 2 seconds of stimulus presentation. It's a rate or a temporal code. Okay, good. So here we have an example of a temporal code. Now, why? Because think about it, the whole 2000 milliseconds, right? The whole 2 seconds, the stimulus per se, right? The variable here in the stimulus didn't change. So the coherence always stays at 0, right? But what is really obvious here that these cells, right, have some temporal profile that you could easily take and, and decode the information of 0% coherence from these neural recordings. Versus if you go to the very top, when all these dots are aligned and go into the same direction, this temporal profile completely disappears, right? Now, of course, the world is not black and white. You see that probably also in the top row, right? The average activity, so the firing rate of these neurons, is elevated when all the dots are aligned 100%, right? So if you wanted to, you could probably also call this here a mixture of a temporal and a rate code. Good. Okay. So I would like now to explain you what phase precession is and how it works, because this is also a, one big idea of temporal coding. So when the mouse, and that's what you see on the very top, passes, or goes through a certain pathway, and this, in its environment or in a maze, then what you observe is that you see cell A, right, is firing when the mouse is in the blue spot, and it's somehow firing. What you see is this, I told you about this theta rhythm, right, that's mediated by interneurons in the hippocampus, which is these cells have really fluctuations going up and down in the membrane potential. Then when the mouse is in the blue place field, then you see cell A is firing, and then you see when the mouse is moving through this blue place field, entering the red, at some point there's an overlap, and also the red cell is firing, right? And interestingly here to note in this middle plot is that the red cell always fires in every cycle after the blue cell, right? And then the mouse moves on, then enters the green place field, and then the green cell comes up and fires after the red cell. So there's a certain order, right? And this order is conserved within every cycle, right, in the firing kind of order of these cells. Good. And that's just an interesting note. Then if the mouse sleeps, you find that find these kind of replays, right, within the shortwave ripples. And then what you find is that even during this replays, the order of cell firing is still conserved, which is, I think, pretty cool. But just on a much, much, much smaller, shorter time level, right? So they really fire all cells within a couple of milliseconds. Okay, good. So the other interesting thing to note here is that within, right, even when the mouse goes through, let's say, the blue place field here, right, as I showed you, then what you see is that the relative spiking of the cell that indicates a certain place field shifts, right, shifts with respect to the cycle, depending on where in this place field the mouse is. And this is an experiment also from John O'Keefe, 1993. So the mouse passes here along this linear track, and then you see at some position a place cell is responding, right? Here, maximum firing rate is 37 Hertz. And when you look at this little cutout, then this is basically you see that the cell is first responding at the peak of the cycle of the oscillation, but then while the mouse is moving through this place field, shifts its peak response to be delayed, right, with a certain phase. And that's what you see basically summarized in D, right? So that the phase delay is getting, really depends on the position of the mouse, right? Good. So this is what you could classically think of, a phase encoding, right? So the position, the cell activity, right, might be always the same, but it's really important at what phase in the cycle the cell is responding. This gives you directly the position of the animal. Okay, good. This is another cool example where we can think of, a temporal coding. You can basically think about this as, time to first spike or synchronicity. So here we have an animal is called the barn owl, and the barn owl, goes hunting at night, and the goal is here to localize the sounds that are emitted by prey animals. And you can do that very simple because the sounds that are emitted, right, you have a short delay between the left and the right ear when the signals arrive. This is the stereo effect. And the barn owl is very, very good at this, right, in using, little delays in sound to localize the angle, right, where's the sound coming from? And then you can use the amplitude, the loudness of the sound to figure out how far is this animal away. So the barn owl is using this interaugural time difference basically to locate prey animals. How does this work? So the circuit that the barn owl has in its brain is called a delay line circuit. And that's what you see depicted on the right. The idea is that somehow input from the left ear, right, arrive on the left side. And then basically because the distance, right, to E, to the neuron E is very short, here the input from the left ear will arrive first, and then it also moves up the ladder, and it will arrive latest, right, with a certain delay in the neuron A. Now the same happens, but in reverse for the right ear, which propagates from the top down. So the signal will be very quickly at neuron A, right? But then will take a longer and longer time due to, traveling. It needs to travel along this axon until it arrives at E, right? So these neurons in the middle, A to E, basically represent a delay in these two signals, right? And that's exactly what we need. Once we have this, or the Banhao has this, right, we can do some, easy geometry. Of course, the Banhao doesn't know how to do this, but it has learned it, right? And somewhat implicitly learned it with its brain circuits. If we have this delay line, the delay, right? If we have somehow the sound frequency, the sound speed of sound, then we can easily calculate here, right, the length of A times sinus phi, right? A we know because it's the radius of our brain, or we have learned implicitly, and then we can basically determine the angle, right? Where was the sound coming from? Good. Okay. So I think I would like to make a break here. Matteo, do we do breaks, or do we?
Yeah. We do breaks. Maybe before the break, there are two questions that came in.
Yeah.
So,
or we can move on and then end earlier, whatever you like. Yeah, I'm fine.
No, I think normally we do a break.
Okay. Let's make a break
questions
and then questions. Yeah.
So how do you force a mouse to sleep right after conducting an experiment?
That's very simple. You keep it awake before the experiment. so what we have been doing is we sit, sometimes next to the mouse, and every time it sleeps, we poke it a little, and then it can't sleep, and then it does experiment. An experiment is excited and it gets rewards, and then it sleeps right after the experiment. Yeah. Mice, however, they don't sleep 8 hours, at a time. They sleep usually an hour, then they wake up, and they sleep another hour, and they're night active, so they sleep during the day.
And then another question is, what are the ripples in the sleeping mouse picture? I think it's a few slides back.
Yes. So sharp wave ripples are somehow, nobody really knows why they occur, but they occur in hippocampus, right? The sharp wave ripple is a signature that you see when you stick an electrode in the brain, right? Then you can really measure a local field potential signature of hippocampal activity, and that's basically what you see. And let me go back a couple of slides. Here, right? Yeah. Here. Good. Yes. So the theta signal and the sharp wave ripples are recorded as a local field potential, right? So this is not cell activity. This is kind of what the whole brain area is doing, right? And what you see is in the sharp wave ripples, these high frequency components that somehow occur. And whenever you see those, right, what people have found is that it seems to be a very fast, sequence of these place cells being activated one after the other. Yeah. Is that the question, or kind of?
I think that answers those questions. Okay.
Yeah.
So if it's okay with you, we can reconvene in 10 minutes at 15.
That's good. Perfect. Yeah. I'll be back.
All right. Ciao.
Exploring this, we're writing a review about this, that if you, for example, if you're the Banhao, right, and if you want to, measure very precisely a time difference between something, right, then a temporal code makes totally sense. Then, a rate code would not, allow you to encode this delay very precisely, versus a temporal code would do that much better. Yeah. So there's regions in the brain that use temporal or time difference coding, and it seems to kind of correlate with whenever you need to measure precise time delay. And there's two things that come immediately to my mind where you need to do that. First, this example here, and second, when it's about motion estimation, right? You may have heard about the Reichardt detector, which estimates, right, basically the difference. And while the object is here, this pixel is excited, then it moves, right? Then it's here. So you also measure a difference between, excited pixels in your visual field, and then you can estimate motion. And the more precise you want to do that, the better you need to measure the delay between the excitation of neurons, right? Yeah. And for these type of systems, seems to be that some type of, temporal code works better than a rate code.
I think if you guys need a follow-up on any of these questions, just write it in the chat.
Yeah.
If not, let's go.
Yeah. All right, good. So I would like to now dive into the question, right? How do we investigate, or how do we figure out how a neuron encodes external stimulus, right? And this is just a very simple scheme here. We have our stimulus, that's S, and it changes over time, S of t. And then this leads to a certain frequency, firing frequency, a certain rate in the neuron, right? Certain response. Good. Now, here we have, stimulus and firing rates. And this is, I took this from the paper Azuz et al. Is basically just to illustrate you that it's not as straightforward as we would like it to be, right? In this case, stimulus on, neuron is firing, stimulus off, neuron goes off. Here is again this experiment that you've also seen in the movie. There's two different cells on the right, and I marked here the stimulus duration in red, right? And then on the bottom, this little arrow you see when the stimulus starts, right? So first, what you observe, the cell is actually delayed, right? So it's not immediately active. That's quite normal because the stimulus needs, the activity needs some time to propagate to the visual cortex. Second, what you see, for example, in cell 1, right, that the cell is actually first nicely responding, maybe even stable, then it stops firing, then it starts firing again, and stops firing, right? Also, the second cell is variable. It's not that it just has, a mean constant firing rate over time when the stimulus is on, and the stimulus is off, and then it shuts off. Right? Good. So what you find is a lot of this type of variability of cells that encode visual stimuli. Now, where could this come from? And when you think about what factors influence, the final encoding or the neuron firing rate, of course, it's our stimulus, right? That's clear. But there could be many other things, right? Such as here, I just list a couple of those. Synaptic noise, right? When I present the same stimulus 2 times, then due to synaptic failure, right, I may have the information, the excitation not reaching this neuron every now and then I don't spike, or I spike less. There could be experimental confounds. So maybe, there's some, when I show this image, it's a little brighter in general, right? There's some tone going on. Oh no, good. There's some tone going on. So I might, oh dear, good. Okay, I'll leave it. Okay, so there's some tone going on, and the neuron might respond to some other factors that I really don't know and that I haven't measured that introduce a certain amount of variability. Then, of course, I have the biophysical properties of the neuron, right? So many neurons have, for example, when they're strongly activated, you have a strong calcium release from the internal buffers, but at some point, all the calcium is internally released, and I can, not fire as fast as I wish, right? So then the firing rate drops. That could probably happen with these first neuron, right? So the biophysical properties determine really how a neuron can react over a stimulus that is presented for 1 or 2 seconds. Then I have the neural activation function, right? So remember that somehow the stimulus comes in, the neuron depolarizes, at some point it spikes, right? When the spike threshold is reached. Now, this spike threshold is not fixed. It varies over time, right? It also depends, again, on the biophysical properties, but this can lead to somehow inconsistencies in the firing rate if this spiking threshold, for example, changes. And then we have to take into account that the stimuli are always or often high dimensionally in space and time, right? So what does that mean? That is kind of similar, goes in the same line as experimental confounds, right? We never exactly know what features the neuron is responding to. And if we show an image, right, which is a very high dimensional stimulus, then if we show a different image, right, that can also in principle show the same thing, some pixels might be different over time, right, or a little brighter or not, and this might influence the spike rate of this neuron. Last but not least, we should not forget that this neuron here is, it's coming, yeah, good. Oh dear. That the neuron is, of course, integrated in a densely coupled recurrent neural network, right? So everything the neuron does right? It sends out to its neighbors, the neighbors change in firing rate, and then it gets some feedback, right? This might also influence the firing rate of a neuron. Good. But for now, let's, move back a step and think about the most simple kind of possible relation between this stimulus input and the firing rate. And the most simple relation is basically just a linear filter that you could basically write like this, right? So we have some factor f, right? That may also depend on the time. Then we multiply it times s of t, and this gives us our firing rate, right? And we can do this, write it like this. If we think about, if we want, it's really delayed here. Okay, good. If we say that there's some delay between the stimulus input and when, the cell reacts, tau would be our delay. Good. So we can write this as a linear filter. Now coming up here, right? So R of t is basically the sum over all the stimuli that we have multiplied with a linear factor, right? And this is a very simple view. This doesn't take into account any nonlinearity or anything in the neuron. Good. So when we think about f, right, our factor can obviously also depend on t, right? And this is possibly, for example, taking into account or representing our biophysiological properties. Then in principle, what we do is, right, we convolve the stimulus input with this f, and this gives us somehow the firing rate R of t, right? So we have a linear temporal filter, and of course we want to sum over all, different time points and stimuli that give us, that give input to the cell. Good. Okay, and this is just the kind of linear version, the continuous version of this. So you can either sum over all time points or you can integrate over time. Okay, so I would like to give you now a simple example of, what type of filter you can implement through a neuron. Okay, here it comes. So basically, when we have the stimulus time point, right, the stimulus on the left, I can take, one time point, and this time point is marked here with this red arrow. Then I can go back in time, n points. Then I would just average over these n time points. I would normalize, right, by 1 over n, and then I would plot, the result at this point in time as output rate R of t, right? So this is known as basically a running average filter, right? So something very simple I could implement with a neuron. Good. Another thing I could do is do a leaky average filter. It's the same idea, right? I'll take one point in time, S of t, and then I'll, convolve this basically with this decay, right? That's the idea of a linear average filter, that somehow points that are far out in time, that have, happened a long time ago, are weighted less and less versus, points or stimulus points in time that just have happened count more towards or affect more the actual firing rate. And then you get a firing rate that looks something like this. Good. Okay. So we talked about somehow temporal filters, right? But also it's very important that we talk about spatial filters. And I'm clicking the slides here, but they are delayed. There they go. Okay. So many of you know that the output of the retinal ganglion cell is a spatial filter that looks like this on the left, right? It's called the surround suppression filter, where you have the pixels in the middle, right? And this is just a small, receptive field within your whole field of view. The pixels in the middle would be, in the middle you have very bright pixels, and then in surrounding you have very dark pixels. Then when you look in the primary visual cortex, what you see is also, kind of these edge filters we discussed before, right? They're kind of elongated. And now the question is, how would we formalize this by the means of a cell filter, right? What a cell does. And in principle, we can take the exact same approach. We basically describe it as a linear filter, right? But now here comes the difference. We don't integrate over time, we integrate over space, right? So you can think about this, the firing rate here, right, is the sum over all pixels from my field of view, right? And I multiply, right, a sum over all the pixels, that's my as x minus x dash, y minus y dash. Then I multiply with my spatial filter, right? And the spatial filters you exactly see on top. Good. So how can I think about this to make this more intuitive? This is my visual field, right? And then I can take a pixel here, x dash and y dash, and then I take all the pixels, right? I multiply with the filter. The filter here you see in red, and then I add this up, right? Good. So I go, along this field of view, I multiply, I add this up. Okay, good. And then at every pixel, right, I multiply what you see here in the formula with the filter, right? And the filter here, I draw it for simplicity, is basically 0, right? Except everywhere in this donut here, right? In the red space, it's, maybe even minus. In the white, heart of the donut, it's some plus value, right? So for this cell, right, only if I'm in this donut here, my pixels actually count something, right? Either positive or negative, and contribute to the spike rate of the neuron. Okay, good. This is our spatial filter here. This is what I draw in red. Okay.
There's a question on a ganglion cell. So I don't understand this ganglion cell. Is this the retina with the fovea in the center, or only a cell?
So the ganglion cell is the output cell of the retina, basically of the retina, right? And I think you actually have ganglion cell everywhere in the retina, not only in the fovea. Just you have only, I think, you have only color vision in the fovea, right? And because you have rods and cones so dense, you have a much better spatial resolution in the fovea than in the rest of the retina. But to my understanding, ganglion cells are everywhere in the retina. Just yes, and the main difference of ganglion cells is that they are the first cells which really send out spikes, right? So they encode some type of analog signal into spikes. Okay, good. So I'll move on here. Okay, so I would like to talk a little bit more about, this surround suppression. This is actually related here. This is what kind of the ganglion cell would send out into the brain, right? And the surround suppression idea is that you have almost a function like this, right? So you have a very positive weighting, right, of the pixels that are in the center for this, receptive field. And then you have a negative somehow weighting in the surround. Okay, good. And in graphics, this is actually a very common filter, right, that you can easily generate if you, for example, subtract 2 Gaussians, right? It's called a different of Gaussians filter. Then you get this surround suppression kind of shape. Good. So let's now look at what would this filter do if we, for example, apply it to an image, right? This basically tells it, what information does what information does the retina send out into the brain? So here I have an image from the Taj Mahal, and then I apply this little filter here, right? I apply it all over again on this image, and what I'll get is something like this, right? And for me, this is pretty cool, right? This is exactly how you can think of the information that's encoded at the level of the ganglion cell that's then sent into the retina. Then the retina, you could do in principle, the same thing again and figure out what the image or how it is encoded on the level of the primary visual cortex. Good. And what you see here is that all the edges are kept, right? Which is pretty cool. I think all the most important information to really recognize what's in the image is still kept here. Good. And so the last step, before we move on to population coding and other ideas is, of course, you can combine those two, right? You need to think of a neuron as combining a temporal and a spatial filtering kernel, right? And then you would basically write it like this, right? You would not only integrate over space, you would also integrate over time, right? If you think about a movie or something, right? Good. And this is actually what's happening. So that maybe explains, right, where at the very beginning, our cat, right, was this neural firing rate was changing all the time because also we integrate over time and we have a visual stimulus. Okay, good. So something that we have forgot today to this point in time, right, is that somehow we have made a very simplifying assumption, namely that the neuron just implements a linear filter. What we have forgotten is that the neuron obviously implements a nonlinearity, right? You have no output of the neuron until the spiking threshold is crossed, and then the spiking frequency increases, right? And to think about what type of nonlinearity actually makes sense, I would like to come up with these 2 questions. the first question is, right, and I give you 5 seconds to think about this. Can spike rates actually be negative, right? Does it even make sense? Do we want that? And the second question is, what happens, right, if the stimulus becomes stronger and stronger? Do we want somehow, right, do we want that the firing rates of the neurons increase and increase indefinitely? Good. Oh no, it's not really working perfectly. Good. All right, good. Here's a solution. Okay, good. So the idea is, of course, that we don't want both of these, right? Negative spike rates don't make sense, and we also don't want to, neurons to grow independently. So what people show, what people have observed in cortical and other brain areas, and what people also used for a long time in machine learning is a nonlinear input-output function here declared as G that has this sigmoidal shape, right? So basically on the y-axis here, you have the output, right? And on the x-axis, you have your filter multiplied with your stimulus, right? Integrating over time and space. And then this is the input to the nonlinearity. If this is very small, we have no output. Then we have some sensitive region, right? We have some output, and then it saturates at 1. Good. Okay. Okay, so we have a linear filter, right, plus the nonlinearity G, and this really determines our output. Okay, so I have a little quiz here for you. Okay, good. So first question, I may give you both questions, and I'll go through, and then I'll let you a couple of seconds, and then I'll resolve this. So since this is a linear system, which of the following is always true? The weighting coefficients always lie on a straight line. If you scale the input by a constant, the output will be scaled by a constant. The output of the sum of different inputs is equal to the sum of the outputs of each of the individual inputs. And the linear filter is a linear function of the input. Good. Which of these are true? The second question is, which of the following inputs might cause a linear system with a positive filter to predict a negative rate? Right? Is that even possible? An input that slowly varies between a large positive value and a large negative value, a positive input that decays to 0 over time, positive input with a discontinuity, and none of these. Good. I'll give you 1 minute and I'll resolve these questions.
Perfect. Before you start again, there are 2 questions, but let's address them after you give the
Let me
answer.
go through the answers here, and then we can do the questions after. Otherwise, it's too much of a mix-up. We need the Jeopardy music or something. So, okay, good. Let me resolve it. So for the first question, 2 and 3 are correct, right? Of course, the scaling for the input and output is the same, and the output of the sum of different inputs is equal to the sum of the outputs of each of the individual inputs. That's what kind of a linear system does or how it behaves. And for the second question, number 1 is only correct, right? Yeah, good. You only get negative values if you have, if the input varies, right, between large positive and large negative values. Okay, good. So I go to the next slide. Do we have questions so far?
Yes, so we have 2 questions. So is there a biophysical limitation to what filter the neuron can mimic?
To what filter the neuron can mimic?
So maybe I can rephrase it if I understood it correctly. Is there a biophysical limit to the transfer function?
Sure. I mean, if you just think about super high firing rates, right? So the neuron cannot, an exponential filter or something like this, right? We always have some limit at, some maximum spike rate, right? So the neuron can spike with probably a couple hundred hertz, but that's the absolute maximum, right? So you cannot go higher. So there's some intrinsic limitation of the output, and that limits, obviously, kind of the transfer function that the neuron can implement.
So linear spatial filtering gives us the borders in the image. How do we account for color?
Yeah, so You have color filters. Very simple. You have edge filters and then you have color filters, right? This is just another kind of route how information is transformed, how information is transmitted and goes along into the brain. Yeah, very simple answer.
And then I think the final one, can you elaborate on the solution of question 2? I think question 2 of the quiz you just gave.
Good. So our, come on, good. Okay, good. Coming again.
Almost.
There. Yeah. So 1 is correct, right? To predict a negative rate. So here, of course, if we have a the nonlinear input-output function, right, we can in principle never get a negative rate. But if we just have a linear system, right, with a positive filter, if we have large negative input values, then we could in principle push this to negative output. Yeah. Yeah. So I think for me, it's almost trivial. So yeah. Yeah.
So there's a follow-up. Maybe more than just limiting timescales, there are certain shapes of transfer functions that the neurons can't approximate well.
Yeah, there's obviously, I mean, if you want to have some type of, step function or something, right? Just doesn't work biophysically. Yeah.
But also keep in mind that the solution to this question is can also be seen as a very biophysical question, right? All the processes inside and how do you weigh the dendritic inputs. So there are different levels of abstraction to the answer.
But there is a certain level of, for example, how fast a neuron can change, right? And it's pretty slow. It has some inertia, right? And there's also certain limitations in the maximum output of the neuron, right? And there's also certain limitations in how fine grain, differences can be really amplified by the neuron, right? So there are a lot of limitations. I think I would be far from saying that any type of filter function can be implemented by a neuron. Yeah.
And there's also another follow-up on the second quiz. The positive and negative value are of rate or amplitude?
This is the amplitude value, right? It's the amplitude. So this is what goes into the filter, right? An input, right? This is our stimulus amplitude. Yeah.
Also, why in the first question is 4 not correct? Sorry, we got a lively conversation now.
Good. Just need to. Okay. Oh dear, this clicker here drives me crazy. Second.
So in the first question, number 4, the filter is a linear function of the input.
Why it's false?
The filter is a linear. No, that's simply not true. No. It's a linear function that takes the input, right?
Yeah, it doesn't depend on the input at all.
It's maybe a fine detail, but yeah.
I agree.
All right, good. Let's move on. Let's not get too long to hang up with these questions. Okay, so problem is, right, that somehow when we measure the activity of a neuron, the output activity, and we measure the stimulus, then these two things are usually not known, right? We don't know kind of the filter function of the neuron, and we also don't know somehow how the nonlinearity is shaped so that it transforms the kind of the output of the filter into a spiking signal. Good. Okay. And what we can do, right? What we can do to figure out basically this input-output function is for temporal features. We can, for example, take our temporal stimulus and then make many, many different features out of this, right? We can basically treat each time point as a feature, right? As 1, as 2, and so on and so on. And we could do it, of course, in a similar manner if we have spatial features, right? We could basically have many spatial features. We may would average a little bit over time and then call, spatial feature 1 the average of, you know, this little red bar, S2, S3, and so on and so on. And of course, what I'm interested then, right, and this is my input output function, is how is this high dimensional representation of data, right, my features, how does this result in a low dimensional firing rate, right? That's kind of the output of the neuron. And here I would like to, you know, yes, that's kind of the description again. Okay, and here I would like to make a point and talk a little bit about population versus single neuron coding, because I think this is a question that maybe should be more answered at the population level and not only at the single level, because I think, you know, information is always encoded by multiple neurons, never by a single one. So we talked about up to now, right, how a complex stimulus, you know, drives a neuron to spike, but what the field now is slowly merging towards is asking how, for example, is a behavior or a stimulus, right? That's also what we do. How does a complete neural population encode this? And this is a little image here. I hope you can somehow see it now with blinking neurons. This is from what we actually collected in the lab using calcium imaging. And what we do is we don't only measure one neuron, we measure many, many neurons at the same time. Good. And there's a couple of different methods to do this, right? This is just a very brief recap. So the classical method was, you know, tetrad recordings. With tetrad recordings, you can, in the best case, record 20 neurons, usually just a handful. There's a new kind of technology, it's called the neural pixel probe. You see it here on the left. It's basically a silicon shank with a lot of electrodes, right? These electrode recording sites. And then it has some type of base, what you see on the right in this image, a cable, and it goes to a head stage and a preamplifier. And with this technology, you can record a couple of hundred neurons in the mouse or in the rat brain. And then, of course, we have, I'll show this in a second, calcium imaging methods that also can, you know, achieve this level of neural recordings. On the right, you see basically how technology really developed and how it now makes possible to record, you know, up to a couple 100,000 neurons at the same time in behaving animals, be it rats or mice or primates, doing some sort of task, learning, behaving, and so on and so on. Good. And if we follow this line, right, in a couple of years, we can record from even more neurons, maybe 1 million or something. Good. This is from my lab, just to show you what we do, right? So here we re-image almost the right half, complete right half of a mouse brain. In this movie here, you see these tiny blinking dots. This is the neurons, right, when they're active, and then you see all the blood vessels. And we can measure this, right, and then ask the question, of course, how is a stimulus or how is behavior represented by the activity, not only of a single neuron, by, you know, 10,000s of neurons, right? Okay. This is another technology that we have in the lab, and I'm done with my technology part, which is called miniaturized microscope. So we basically place a small 2-gram miniaturized microscope on the head of the mouse. It runs around with it. And then we see kind of the activity on the neurons on the right. And you can already kind of grab or gasp from this movie here that it's not so easy to really just say ad hoc, right? What is, you know, what the mouse is doing on the left, right? How is this encoded by the neural activity on the right? It's not so straightforward. Good. Okay. Okay, good. So again, we would have these, you know, two, let's say, high dimensional, right, features that we have, or two stimuli. And then we have two features. We may have, you know, different filtering probabilities, different filters for these two stimuli. And then we sum these up, right, the result of these filters. And then we put it into our input-output function. And then we have an activity of the neuron, the output activity. And here now we're interested in how does these different stimuli input, how does behavior encode, is encoded by a whole population, not only by a single neuron. Good. So we focus on this. Population coding, this is just the definition. Population coding refers to information available from an ensemble, right, that goes beyond the simple summation of individual signals, right? This is quite important. So when is, you know, you could say, well, I can just look at, you know, the individual neurons, right, and could just then sum them up and all their firing rates, right? And then determine how likely a stimulus is. But that's not so easy. Keep in mind that neurons, for example, are highly variable, right? Think about the hypothetical case, all the neurons would encode exactly the same, right? Many neurons, 10 neurons, they would be highly correlated. You won't gain any more information from taking into account more neurons, right? One neuron would be enough. Versus if neurons are highly decorrelated, right? Then and are noisy, right? Then it's beneficial to look at more neurons. To train your decoder, for example, with more neurons, you get a much better performance in predicting behavior or stimuli. Yeah, that's just a little side note. Okay, good. So what do we do to kind of extract or determine these input-output function, right? Either be also stimulus somehow encoded by a single neuron or by a population. So the idea is that we take our stimulus, right? This is a temporal varying stimulus. We divide it in temporal features, and then we can plot these temporal features basically in high dimensional space for every time we present the stimulus, right? Good. This is your first example, and then what I need to do is now we need to repeat, right? We need to kind of give the stimulus again and again and again and again. And what I want to get out of this, right, is the probability of the response given a certain stimulus, right? That's kind of my my encoding or my mapping. Okay, so how do I do this? I repeat, right? So I get more stimuli, more data points here. I repeat again, right? Okay, good. And then I get something like this, right? Great. And then I'm either in the situation that somehow, you know, I know exactly, okay, good. I know, or I don't know any labels, right? I just have collected these neural activity recordings, but I don't know any behavior. I haven't recorded any stimuli, right? So then I could do some unsupervised learning, and we'll come to this in a second, how I could do this. If I'm lucky, I have somehow recorded exactly the time, right, that I presented the stimulus. So I have a label, and I also recorded, for example, the cell's activity, right? Good. So let's start with the supervised approach, just because it's much simpler. If I have some label, for example, in the blue dots, right, I had a spike, and the red dots, I didn't have a spike, right? Then it's very simple. I have basically two of these populations. I could just basically determine their means here, and then draw an arrow or a line, right, through these two means. And then I would just project these whole population response from the high dimensional space into one dimension, right? And then what I get, for this case, for example, is this bimodal distribution, right? Good. Then what I would do is, right, I would define some type of I would learn a classifier, right? I would define some type of decision boundary where I say, well, all the responses, right, from left from this classifier, good. Okay, all the responses left from this classifier are basically non-spike, right? Versus all the responses right from this classifier line here are a spike. Good. So once I've trained this classifier, it becomes very simple to decode or to encode any new incoming stimulus, right? So think about it. Now I have a new stimulus here. Zoom is still late here. Good. I have a new stimulus here. Now what I do is I project it down, right, to the dimension of my encoder, right, dimension that I have previously found. And then I can, you know, look at my bimodal distribution, my decision boundary, and figure out, is this new data, right? Is this new data point, is it on the right of this decision boundary or on the left? And I just normalize it to 1 for, you know, simplicity. And when it's on the right, I say, well, this is a data point that should elicit a spike of this neuron, right? So what I've done here, basically, I have figured out somehow, right, the input-output function of a neuron, right? I've wrote a decoder that tells me, well, this stimulus elicits a spike and the other stimulus does not elicit a spike. And this is a very simple, you know, one-dimension linear decoder here. Good. So how do we find the input-output function for a single neuron? As I said, these two things, you know, the filter and the nonlinear input-output function are not known. And the other way how I could do this, right, is, I'll show you this, is through thinking from a probabilistic perspective, right? So what I want is, I want the probability of a stimulus of a spike given a stimulus, right? This is my input-output function. And S1, right, as identified in this case by our linear classifier. Good. So how would I do this? We can find the input-output function, for example, from data using the Bayes rule, not repeating kind of, again, Bayes rule. So we have here, a short flash of Bayes rule. We have a posterior probability of the stimulus given the response. The likelihood is the probability of the response given the stimulus, and our prior, right? How often we think the stimulus in generally occurs, right? And then basically divided by the probability of the response. Good. So how can we now get our P spike of S1, right? This is basically plugged in our, what we want into the Bayes rule. And then what we can do is something like this, right? We can basically measure the probability, we measure the spikes, right? And we measure the probability of S1 to occur. And then we can basically take all, the times where we had a probability, we had a spike, and we can take the probability of PS of 1. Then what we can do is if we have all these points, we can basically generate something like a histogram, right? Or here on the y-axis, we have the probability. And what you see is that, again, it looks like this, bimodal distribution. Of course, it always doesn't look that nice, but in principle, right, the first thing would be, right, our first peak would be the probability of a spike, and our second peak would be the probability of, the first peak would be probability of stimulus, and the second one, probability of S1 given a spike. And then what I can do again, right, I have this probability distributions define some type of, decision boundary and train my classifier. Good. So this is what I want to get out, my probability of a spike given my stimulus, right? And I can now calculate this, right, when I have this probability distribution that I showed you in the last slide. And then just to, think about it, if I have the probability of the stimulus is exactly the same as the probability stimulus of a stimulus given a spike, then they have absolutely no relation, right? That's clear, right? So because the stimulus does not at all influence, the spike does not at all influence this probability of a stimulus to occur. The other way, if this is different, right, if the probability of a stimulus given a spike is different, then somehow I'll see that these peaks, right, are really shifted. And the distance of the peak is really kind of, determines basically how selective, right, is the cell, how selective is the spiking with respect to the stimulus input, right? You can even measure this distance. Yeah, good. Okay, good. I would like to talk the last 5 minutes about how can I measure distances in population responses, and how can I basically classify or think about similarities and differences in population responses. So here we basically have 2 neurons, and these 2 neurons are this is simple population, right? It's only 2 neurons. And then if I repeat the stimulus, right? So I have, for example, baseline or I have the stimulus, then I also get these population responses, right? It's the same idea as we had before. And now I want to figure out how similar or how distant these populations are, or I want to train a decoder. Then what I'll do again is I somehow connect their means here, right? And then I project everything down to 1 dimension, and then I can, for example, calculate the peak-to-peak distance. This would be the same as calculating the Euclidean distance between the means of these populations here. Good. Okay. What I argued, for example, in my last paper was that keep in mind that this is a very simple approach. Taking the Euclidean distance, that's also what a linear classifier does. What it does not take into account is that the variability, right, of these population data, be it our temporal features or be it our neuron features or our space features, can have a different variance in the different data space dimensions, right? It doesn't need to be unity variance in all different directions. And then what you see on the left, taking the Euclidean distance in principle and then drawing a decision boundary, right? Think about how this would look here. Doesn't really make 100% sense, right? To draw it somehow like this here, right? What I want in principle is something that looks like this. Okay, good. So how do I get this is what I have to do is I can normalize somehow, or I can my variability. And this is what the Mahalanobis distance does. It multiplies with the inverse of the covariance matrix. This is here our multiplication with the sigma, right, -1. And then what we do is by we transform, right, our data into another subspace of unity variance, right? Then our population clouds are nice and round, right? And then we can just take the difference between the means and train our classifier or do other things about. Good. This is just an overview slide that if you do these types of encodings and classifications, keep in mind that somehow two things are important, right? First is the difference in the means of two population responses, right? And that's the top row. So if the mean gets bigger, I can nicely classify. I have a very different, I have my neuron is dependent on spiking or not, right? But it also depends on the variability of that I see if I repeat the stimulus, right? You can easily see that on the right, variability is small and I can easily classify. On the left, the variability is super big and I cannot really figure out, does my neuron spike to the stimulus or not, right? So main point here is, when you think about encoding and decoding, two things are important, right? To measure input-output function or to classify population responses is what's the distance of the mean and what's the variability. Okay, good. And this is in principle, when you think about, linear discriminant analysis, it's exactly what happens. So you basically choose, right, a good projection. And in this case, you choose a good projection, which is, looking downward. If you choose a bad projection, right, you will not be able to discriminate, right? And that's exactly what you have to do. And in the ideal case, you take into account that variability in different dimensions might, look different. Good. Okay. Okay, good. Unsupervised, just give me 1 minute. Unsupervised analysis. Of course, you could say, well, we don't have spiking, right? Here, we don't have a label for these data points. Then what you could do is you could look at the direction of maximum variability, right? This would be, for example, this one here or this one. This is what PCA does, right? And then you would just do a projection along these lines, and you might also find this bimodal distribution. And you could even, if it's really bimodal, right, train a classifier and say, on the right, I have my state 1, and on the left, I have my state 2. Yeah. Could even define a decision boundary. Yeah, good. And that's it for the lecture. I have, this is just a funny picture. If you do PCA on faces, then you get eigenfaces. It's like people like me laugh about this stuff, but so all right. So I'll stop here. I think we should not do the quiz. You have the slides, so please write us if you want to know the answer to these questions, and then I'll tell them to Matteo later today, and then he can communicate these. All right, good.
We can go over them at the beginning of our next lecture next week.
Whatever you like. I mean, I'm fine. Yeah.
We'll see. Let's see if they ask.
Okay, good. All right. Yes. So that was the lecture for today. Thank you very much. And then we're ready to take some questions. And then I think we start with the exercise, right?
Yeah, we got
10:15, or when do you plan to start with the exercise?
So we can have a 15-minute break, and then I can go over the exercises of last week.
Yeah, good. And for now, we probably have time for some questions then.
yeah.
There's one question, but it's about the format of the slides. Is it actually uploaded? Yes, the slides
It's
are
PDF, right? Should be PDF.
Sometimes it's not.
No, it's a PDF. Yeah.
No.
Can you open it? I can check. Let me check.
Yeah, I opened it. She can open it.
I can open it as PDF. Yeah, should work. Yeah.
There are some stuff missing. Okay. Okay.
Yeah. The problem is, of course, that I have animations, and that these are not captured in the PDF slides. I guess that's the benefit of coming to the lecture. so yeah, I haven't found a solution to this.
Because
but I just went through it, and there are plenty. I mean, 95% of the lectures here, and you have the recording. I think it's fine.
Sure, sure. I mean, so, like the things that pop up, right, that I have as animation, they are not videos, of course. I think the majority of things should be there. I'm just checking the PDF. Yeah, I mean, if you give me some details of what's missing, what I can do is I can try to, take out the animations and make an extra slide or something, and then upload it again. Yeah.
Okay, but
Currently,
yeah,
I don't see what's missing.
of it is there.
Yeah.
All right, so someone actually asked if they can have the solutions of the questions.
I guess.
Someone else asked, these classifier techniques only work when using either rate coding or temporal coding uniquely.
Sorry, give me a second. Yeah, I can just tell you the numbers of the questions, the right questions, or we could go through them, I guess. But yeah. I mean, we can briefly do this. Wait, let's try to do this quickly. Good. Okay, if we discretize a stimulus waveform in time, we can represent it as a vector in some vector space. What is the dimensionality of this vector in space? Good. The standard deviation of the stimulus, the number of presentations of the stimulus, the length of the stimulus in seconds, or the number of points used in the discretization. Give you a couple of seconds. The right answer is number 4. So it's really the points, right, that we use for discretization. Doesn't matter so much how long the stimulus is in seconds, right? The discretization is important because that determines, how many data points we get and how high dimensional we get. Okay, good. Second question, when we plot the probability of a spike given a stimulus as a function of S1, why does probability of a spike only act as a scaling factor rather than as something that changes the general shape of the function? Good. So answer 1 is not correct. P spike has a large effect on the shape of the conditional distribution plotted as a function of S1. P spike influences the spike triggered average. Answer number 2. Answer number 3. P spike is the first moment of the spike conditional distribution. And number 4, P spike is not a function of S1. Right.
There is another question.
Okay. So the answer is, again, number 4 is correct. P spike is not a function of S1. Very simple. Okay, good. I'll go to the last question. Principal component analysis gives us a method to find a representation of a data which has a lower dimensionality, giving us a computationally easier problem to work with. Find the vectors along which the variation of our data is maximal in our feature space. Neither of these, both of these.
Oh,
CA, right? Unsupervised method. We don't have labeled data. Again, number 4 is correct. Nice. Yeah, you got it also in the chat. Perfect.
A lot of people wrote it in the chat.
Yeah, good.
Right. So yeah, there are 2 questions which I'll just pose. So these classifier techniques only work when using either rate coding or temporal coding uniquely.
Yeah, so I mean, they always work. It really depends on how you bring the data into your feature space, right? So your feature could be the rate, the spike in frequency. Your feature could also be the delay, right? Or the phase delay within a cycle, right? Then you can always use supervised or unsupervised classification methods. And
there's another one. I have you mentioned negative spike trains. First, you mentioned that they did not make sense. Then you did a problem about how to obtain them. I don't get it. And
yeah. So negative spiking doesn't make sense, right? The cell can only not spike, right? So And that also relates to the input, to the nonlinearity, right? The nonlinearity is 0 when there is, the membrane potential slightly depolarizes, but also with the membrane potential can actually go more negative, right? But you won't see this in the output of the neuron. So the idea is somehow that the filter function, of course, right, of a neuron, there could be some, negative input signals to the neuron, right? If you have a strong inhibitory negative signals, and the neuron could even reach sometimes minus 100 millivolts, so it could be hyperpolarized a lot. But you will never see this as a negative output because the neuron only produces spikes, right? And it produces this if some membrane potential around 45 millivolts or something is reached, which requires a strong depolarization, right? A strong positive input from the filter. It's very different, right, to artificial networks, right, that basically send out positive and negative. Or they're also only active. So the ReLU function is only positive, right? Just they have negative weights, so this is then transformed into a negative input into the next neuron. All right, good. Do we have another question, Theo?
No, I think.
All right, good.
That's it.
Yeah.
So yeah, let's do a 5-minute break and meet here at 10:15, where we can go over the exercises of last week.
Okay. And I would leave if that's okay.
You're free to go.
Good. Bye-bye, everybody. See you next week for the next lecture.
Bye. Thank you, Benny.
Ciao.
Bye-bye.
Let me resume the recording. Okay, so I'll quickly go through the exercises of last week. This I don't think should take a full hour, but let's see if you guys have questions. Okay, so is it working? Okay, so the first exercise, components of synaptic delay. So in this exercise, we look at some data measured on the giant squid synapse. The plot is adapted from a publication of 81. It showed traces from the presynaptic cell and its postsynaptic cell. And then the first task was to identify these traces A, B, C, D as the postsynaptic current, the presynaptic action potential, the postsynaptic potential, and the presynaptic calcium current. And so we can go through it, and so I would say that the first trace A is the presynaptic action potential, B is the presynaptic calcium current. So if you remember how synapses work, you have an action potential. When the action potential gets to the axon, it will trigger a calcium current, so that there's a time shift. And then we have the postsynaptic current in C, and finally the postsynaptic potential in D. So you can kind of see it as A leads to B, B leads to C, and then C leads to D, kind of a consecutive kind of thing. And then the next question was, what are these delays A and B? And so A is approximately 0.8 milliseconds long, and it comprises of the gating of the presynaptic calcium channels. As we just said, you have an action potential, and this action potential propagates through the axon, and then the synaptic cleft will release calcium. And so this just takes a little bit of time. And what is B? B is approximately 0.2 milliseconds long, and it comprises of all the processes from the calcium current and the subsequent steps. So it's literally the calcium vesicle fusion, transmitter diffusion through the cleft, and then the gating of the postsynaptic ligand-gated channels. Okay, so unfortunately I cannot move screen if I'm still sharing. So if I'm still, okay. So this is exercise 1. exercise 2 was about quantal release. Both exercise 2 and exercise 3 were about quantal release. And so we're designing an experiment to test probabilistic nature of neurotransmitter release. And we know, so we're able in this specific circumstance to make the probability of release very, very low. And then they ask a whole bunch of questions. And so we know that the release probability follows this, this law, Poisson law, and they stimulate the nerve 500 times. What happens? Okay, so what we know is that this is the law that it follows, and we know that the mean is 5. So on average, when I stimulate the synapse, I'll have 5 releases, and that we stimulate the nerve 500 times. Just for completeness, what is a Poisson distribution? So this is a whole family of Poisson distributions as we move the mean. So if I have a very low, so in these exercises, the mean is called M. However, in statistics, in Poisson distributions, the mean is lambda. And so as we move lambda, so for a lambda of 1, you have this kind of black curve, and you can see how the mean and the variance and the shapes change as we increase lambda. And so consider that we're working with a probability of 5, so this blue one here. Maybe it's the next one, I forget. No, it's this blue one. Okay, and so it asks how many failures of transmissions x equals 0 do we expect if we observe 500 trials? And this is basically just plugging in the numbers. So, so if we plug in the numbers, so we have 500 trials times 5, which is the mean elevated to the x that we're interested in, over 0 factorial to the e2 minus 5, and then we get 3.347. And so then the next question is, how often do we expect 2 quanta to be released? And it's exactly the same protocol. And so we just plug in and we get 242. And this should be of no surprise if we go back and look at the, at the shape of this curve. We'll have that the maximum number of occurrences is at 5. And so 0 is pretty low, 2 is the slope on the way up, 5 will be the peak, and then it'll come back down the other side. Yeah, this is, I think, pretty clear and self-explanatory. So number 3, okay, I do the usual thing. Let's go read the exercise. Quantal release. So we have this histogram, and we need to answer a bunch of different questions, right? So these recordings were made under low extracellular calcium and high magnesium concentration. These special conditions permit to reach extremely low release probabilities. And that's exactly what we want if we want to observe these kind of things. Furthermore, we have 200 events, and the mean amplitude of all events is 0.93 millivolts. Okay. So what happens? So the first question is, what do the different peaks represent? And so this question should be answered from kind of the previous exercise as well. So we have failures of transmissions, 1 vesicle, 2 vesicles, and 3 vesicles subsequently. The second question is, from the graph, what is the quantal amplitude? Q. The quantal amplitude, known as quantal size, is the amplitude of the response induced by transmitter from 1 vesicle. And so again, here we can kind of look at the plot, and it's 0.4, because you can just look at the mean of the fitted histogram around B. So B is one release vesicle, and then and that vesicle is of amplitude 0.4. So then using the results from B and the mean amplitude of all events, calculate M, the mean number of vesicles per release. Can you imagine another way of calculating M assuming a Poisson distribution? So here we can calculate it in 2 ways. The first way is, so we have the overall, like the mean amplitude of all events, and we have the amplitude of a single event. And so we just divide them, and it's 2.3. And so on average, you will have 2 something releases, which obviously it's not true, right? So you'll have either 2 or 3 releases. Let's be clear. And then there is also another way of calculating it, in which we know it's a Poisson distribution, and we know and we know that and we can just plug in the numbers, right? So M is what we're looking for, and we just invert this function here to get M, right? Oh, also, because we know the result for 0. So we have this Poisson, we know that for 0 releases, it's 18, because the graph tells us that. And so then knowing all of this, we can invert to get M. Okay? Good. This should not come of no surprise, but yeah, if you have questions. Okay, and number 4, assume that M and Q are measured for a given experiment. In a subsequent measurement in which experimental parameters have changed, M and Q are measured again, and they might have changed. How can you detect an increase in the release probability? How can you detect postsynaptic modifications? Okay, so so presynaptic changes increase the value of M, whereas the value of Q would remain the same because Q is a property of the postsynaptic neuron, right? Because while the postsynaptic changes would leave M unchanged and would increase Q. And so these are the 2 parameters. Any questions?
Yes, there is
Yes.
a question. Should in C both ways do not give the same result, the calculations?
That is true. Because we are, I mean, so that is very true. That is because in the first way, we fitted the curve in B not very precisely, right? I mean, you can see it here. Yeah, we can kind of fit it. Okay, 4 is the midpoint. Let's say 0.4. But I mean, if we were to do it with actually a statistical model, probably the result would be much closer to 2.41.
And they are, reasonably close.
They are exactly. So I mean, you could think that if, yeah, they're reasonably close. I mean, science. I mean,
That's what you said. It's about the fitting.
Yeah. Okay. Okay. We went through all of this. Perfect. So exercise 4. So it was called Back to the Basics. And it was just draw an action potential on a voltage time graph. On the time axis indicate where the sodium and potassium's channels open and close, and the reversal potential and the threshold. And so this is basically the plot you're looking for. So we have a depolarization, repolarization, hyperpolarization. We have the resting potential of minus 65 millivolts. We have the threshold theta. We have the reversal potentials of the sodium and the potassium. The sodium is more positive. The reversal potential of the sodium is obviously more positive than the peak, and the reversal potential of the potassium is slightly more negative than the resting state, and it's closer to the hyperpolarization point. And this should come up as of no surprise, given the fact that we're now 7 weeks into this course. But yeah. And so the last exercise is influence of ion concentrations. And so we have this plot here. Assume that we have a cell whose membrane is permeable only to K+ and calcium ions, so potassium and calcium. Such a cell shows a resting potential and an action potential, like in the figure. If one varies the concentration of extracellular potassium K or the concentration of extracellular calcium, one observes the following changes, and those are the plots in B. Let's answer the following questions. And so, so which of the following statements about permeability is true at resting state? So to answer this, I won't go back to the slide, but I mean, I'll put online the slides. So we have that at resting state. We have that the changes in the concentration of potassium will affect the resting potential much more than the action potential, and vice versa. Changes in calcium concentration will affect the action potential more than the resting potential. And so at resting state, we have that the permeability of calcium is higher than the permeability to calcium because of what I just said. And vice versa, during the action potential phase, we have the other way around, in which calcium is more permeable than potassium. Compare K in and K out. Compare also calcium in, calcium out. In which direction do more ions diffuse to? What is the resulting current flow? Discuss how potassium and calcium currents relate to each other. So, okay, so this is what we just talked. And if you want a more analytical solution, then you can look at the Hodgman-Huxton-Katz equation, just considering potassium and calcium, right? I mean, you ignore sodium and you can do this. And chloride, yeah. So which of the following, okay, this we went through. Okay, so if we have that the potassium in is greater than the potassium out, ion channels will have the tendency to flow outwards. And that the permeability to calcium is negligible. So, so yeah. And finally, when the cell is perturbed mechanically, the membrane transiently hyperpolarizes. What permeability changes might be responsible? What ionic current typically leads to hyperpolarization at physiological concentrations? Sorry, I got tongue-tied. Again, since potassium dominates at resting potential, it's going to be potassium. And yeah, so these were the exercises for this week. I think they were kind of a nice overview of a lot of synaptic concepts. And yeah, if any more questions? No?
Nothing.
Did I go too quickly? Maybe.
It was okay. I mean, it was.
I mean, if there are no questions, then I don't know. I mean, we're a bit puzzled because we've seen a steady drop in the number of submissions of the homeworks.
There was a question.
Okay.
Could you repeat how to obtain A, B, C, D in question 3?
Okay, question 3. Okay, so A, B, C, D are just these points here in this histogram. And so for and the question is, what are these A, B, C, D? What do they correspond to? And so given the fact that we're looking at, it's a plot of synaptic efficiency, and so that we have that for A, we have an amplitude of 0 millivolts at the end plate potential, and this happened 18 times. And then for B, we have an amplitude of 0.4, and this happened 20 times, and C, it's 0.8, and D is 1.2. So we can kind of conclude that for A, it's a failure to transmit. For B, it's one vesicle release. C is a 2 vesicle release, and D is a 3 vesicle release. You obviously have to keep in mind that the only exact, say, science number here is 0 or A, because so if there's no failure if there's a failure to transmit, there will be no amplitude on the end plate potential. If there is one release probability, if there is one vesicle released, this release will have a mean, which is 0.4, but will also have a slight variance, right? And so then if there are 2 vesicles released, the variance will grow a little larger. And for 4 vesicle releases, it'll grow even further, and so on and so forth. And so this is why we need to run these experiments with a very low calcium concentration, high magnesium concentration, to try and make the vesicle transmission as sparse as possible, because if we were in normal conditions, and this would lead to many vesicles being released, then all of these curves would average out. More questions?
And when does the number of events decrease when the amplitude increases?
Because so the number of events increase because okay, so there are two answers to this. The first one is so for let's take scenario D. So we're looking at all events that have a release of 3 vesicles, and so we have that it'll be the bar D where it's labeled, but also probably the bar to the right and to the left will be included in D. So what one should take is you should fit these events to a normal curve and then take the integral to that. And probably the integrals would be very, very similar. However, there's also a more scientific answer, which is maybe in these conditions, the probability of releasing only one is, we are in the conditions where the probability of releasing only one dominates. So if we were to increase the extracellular calcium, then, you know, this curve would move more to the right. And so maybe B would be lower and D would be higher. So there are two ways you can look at it. More questions?
Yes, for 7.4, where sodium and potassium channels are open and closed.
So 7.4, so we know that the beginning, so this is an action potential event. So the resting potential is at -65. And so we know that at the beginning of the event, we have that the sodium tries reaching its reversal potential, which is up here. And so in the beginning, it'll be sodium channels, and then potassium is in charge of repolarization and hyperpolarization. I strongly advise you to go and look at the exercises of last week and maybe the week before as well, in which we've covered this extensively. And I think this is a very fundamental concept.
And where are the reversal potentials of sodium and potassium in the same figure?
So potassium is K, so EK is the reversal potential for potassium, and Na is sodium, and so the reversal potential sodium is up here. So I don't have I mean, if we're doing an analytical exercise, we could look up the exact values. In this case, we just need to know that the sodium reversal potential is positive, and it's above the peak of the action potential, and the reversal potential for potassium is negative, and it's below the resting potential.
So someone is saying sorry that she forgot to put the exercise there.
Oh, no, don't worry. If you did them, I'm very happy. I'm just afraid that we've lost 3/4 of the class. And so yeah, I mean, the exercises that get uploaded are very, very good. I checked the few ones of this week, and I mean, yeah.
Okay. So I guess there's no more questions.
Okay, so Carla started this tradition to kind of stop sharing and just hang here for 5 minutes in case questions come up. So I say we do that, and so I will stop sharing. We will pause the recording, and